Welcome to the AI for Coral Reef Challenge final presentations. For the people who don't know us yet from Fruit Punch AI, we are an organization that organizes AI for good challenges, and this entails that we crowdsource AI solutions in order to solve the problems gearing towards the sustainable development goals. And for this challenge, we collaborated with Reef Support, and together we are trying to solve or create AI solutions to detect and classify coral reef to aid the conservation and research of life underwater. They will tell you a little bit more on what that exactly means. But I will show a little bit of how we organized this challenge. So 2 months ago, we started by looking for people who are AI enthusiasts, people with a desire to learn more about AI or to implement AI in a way that actually helps the world forward. Because many of you will already know this, but for the people who haven't been able to follow this challenge as close as we did, the reason why we started this challenge is because as of this year, since 1995, already 50% of the global corals has bleached or has already died. This is a huge problem since 30% of marine life and also 1 billion people on Earth are depending on these coral reefs. So it is essential that we are building systems to map where aid for these coral reefs is most necessary, so we can advise and build policy towards conserving these coral reefs. What I have today for you is the final presentations from this 10-week challenge. First, we will go into a short introduction from Marcel and Johan from Reef Support. And then we have the presentations, firstly from Jaka from the Automated Color Enhancement group. They worked toward creating an AI capable of color enhancement. Then we will head on to the presentation from Max, and he has been busy with object segmentation and especially with calculating benthic coverage. This is essential in order to create good policy to aid the conservation of coral reefs. And last but not least, we have Leonard, who will tell us everything that he worked on towards classifying coral types. And finally, me and the people from Reef Support will do some closing words. But first, I'd like to give the floor to Marcel and Johan for a
yes, short... yeah.
Okay, I stop sharing. And all right, go ahead.
Yeah, thank you very much, Dorian, for the amazing introduction. So hi everyone, my name is Johan Runner. I think I've met most of you, but I do see some new names in the chat. So thank you all for being here. So I'm the CTO and co-founder of Team Reef Support. We started off Reef Support a year and a half ago with Marcel, and our mission is to enhance marine biodiversity and strengthen the blue economy through innovation. So why we're doing this is although coral reefs only cover 1% of our ocean's floor, they support over a quarter percent of our marine life, and over 1 billion people directly depend on reefs for food, income, and protection. Because something that you might notice that reefs serve as a perfect natural barrier against pounding ocean waves. But unfortunately enough, we've already lost or severely damaged more than 50% of our reefs worldwide. And studies project that all reefs will be at risk by 2050 unless action is taken now to reduce these threats. Fortunately enough, we have dedicated scientists that are working hard every day to continuously monitor these reefs. But although we have over 16,000 designated marine protected areas globally, only 1,000 of them are fully protected, and that is because reef monitoring today is cost, scale, and time expensive. So our mission at Reef Support is through the use of AI in combination with emerging satellite technology is to provide accurate, standardized, and actionable data to help monitor and protect our reefs. because we believe that by combining commonly used underwater imagery in monitoring with automated image annotation, we can drastically improve how monitoring of coral reefs is done worldwide, particularly in terms of allocating those limited resources. So we actually came across Foodbench a long time ago on LinkedIn when they were getting started with a couple of their very interesting challenges. I believe it was the AI for wildlife challenge at the time, and also the AI for health, but it might be another one. I might be mistaking the timeframes there. But we believe that by partnering up with them, we can propose an awesome challenge for AI enthusiasts out there using the data sources that we have collected over the time frame that we support exists by our partners and open source data sets as well. And so we believe that by creating this Coral AI challenge, we could leverage the help of AI enthusiasts worldwide to help us with our mission. So yeah, after a month and a half of hard work and planning, thanks for that, Sako, Dorian, and Busters. The challenge finally kicked off, and after 2 months of hard work, we're finally here. So I'm very enthusiastic to hear all of the hard work you guys have put in. The final presentation, I'm very excited to hear everything, and yeah, good luck with your presentations. Thank you all for being here.
Thank you, Johan. Yes, we're all very excited. So let's get started. Jaka, I would like to give you the floor first to tell us more about what you reached in the automated color enhancement.
All right, hello everyone. My name is Jaka. I'll be presenting the work we did in our group. Can you just confirm that you can see my screen, full screen?
We see it.
Great. So we've been working on automated color enhancement. And here is our team, Harriet, and me, Jaka. And as you can probably see, those images are very old. So just to establish that we are indeed enhancement experts, here we go, new images. So we wanted to do the same for corals. And the problem here is that when you take underwater images, you quickly lose contrast and color because of the diminishing light that gets through the thick water. And this leads to a decreased performance of AI approaches for segmentation, classification, and other tasks, which rely on contrast and color a lot during their inference. So, of course, our mission was to restore the contrast and color using state-of-the-art AI approaches. And we set out to find the correct approach to do so, which turned out not to be such an easy task, since many of such approaches use some additional tools on images or use certain additional sensory information, such as depth, to infer the final result. And on the right-hand side, you can see one such approach, which is see-through, and it uses some additional tools and also some depth information to make this beautiful result. So we experimented with multiple approaches to get to that point as close as possible without the need for additional sensory information or markers. We started first with image processing, for example, PLAHE, which is contrast enhancement technique, and similar, which was the most basic. Then we started, then we looked into underwater CNNs, which are basic neural networks for the task, and so on and so on. And I will present these approaches one by one. So first, image processing. We tried a few techniques, and as you can see on the bottom image, on the left we have input image, on the middle we have some custom adjustments to the histograms of the image to make sure that it is indeed possible to restore color from them. And on the right-hand side, you can see our results from image processing, which you can see that they are quite nice, but not there just yet. So there is still not enough color there. And also the contrast is either exaggerated in some places or missing in some other ones. Then we looked into generative adversarial network-based approaches, which are a collection of approaches that are very good at transferring one style onto images. So for example, you can pick a colorized underwater image and try to transfer that same style to new images, which sounded quite promising. And actually there is a huge base of research for GAN-based approaches. But as we found out on our local tests is that there is really not enough, how to say, global image coverage of the approach. So as you can see on the right image, it focused more on the center while it left out the outer regions. And it also just tries to uniformly convert the image, which is not the correct approach. It just tried to make everything a bit more red, which also prompted the now famous response from one of the organizers, it's too red. So we decided to abandon that and went for underwater CNNs. This was a very simple CNN, as you can see on the top right side, basically just a couple of layers, so not very deep either. And we set out to perform a custom training of the model on an underwater data set called Underwater ImageNet. But unfortunately, our attempt failed too. After quite long periods of training, we were still getting just reddish and brownish images. And upon closer inspection of the original paper of the authors, we actually found out that their results were also similarly brownish. For this approach, we actually did a custom PyTorch implementation available there if you're interested to check it out, so that for the future we can maybe still return to it with more data and tackle the problem. And then finally, we found a CNN-based underwater image enhancement technique using two color spaces, or in short, UIEC squared, which uses a slightly more complicated neural network. It is basically split into three separate branches. The first one tackling RGB pixel level information. And second one is an attention layer or branch, which tries to focus certain parts of the propagation on image to pay attention to the colorization. So for example, if you have a fish there, this branch will try to focus on the fish and make it more colorful. And finally, you have the third branch, which is in a different color space, HSV, which tries to globally fine-tune the final colors of the image. And if you think about it, this is actually quite a good approach because it's similar to something that a human might do when restoring images. First, you tackle the histograms, you then focus attention on individual bits to make them prettier, and you try to play with HSV to tune individual bits to match what you're expecting. So we ran a couple of images through with the provided model. And as you can see, it is doing a very good job. On the top right hand side, you can see a very low contrast image being transformed into a beautiful contrasty image, and also already restoring some bluish and yellowish color here. And on the left, low left side, you can see even more color being restored. Same for the lower right. So we decided to continue with this approach, and we went for some custom training. And again, here we used the same data set that we used for underwater CNN, focusing on underwater ImageNet dataset specifically, because we learned that this dataset has more natural images in them, and it approaches more the domain that we were working on. And you can see that after about 40 epochs of custom training on this dataset, we're getting quite good results. On the left hand side here, you can see the input image, a fish, and on the middle, you can see the ground truth, which has very beautiful colors. And on the right hand side, you can see the output of this method, which is restoring color very nicely. Actually, I didn't find one image of a, I don't know how it's called, of this thing that's surrounding the fish in a different perspective. And it's, the method really restored its glowing green. It was quite nice to see. And furthermore, we tackled another challenge, which was trying to make this approach useful for the following teams. So image classification and segmentation, starting with image classification, which was mostly using ATL dataset. We tried to basically use this approach to produce a new color enhanced dataset on the images that were used for image classification. And here you can see some of the colorized or color restored images from that dataset. And we also prepared a brief demo for you because we are quite certain that after the presentation, you will definitely want to give this a try. So if you can still see my screen, here it is. If you will click on that link that I will also paste into the chat, you can basically read a bit about how we tackle the problem. And you can also upload your custom images here, and it will do color enhancement, and you will be amazed at the beautiful coral reefs that you will see. Okay. Sorry. So as a wrap up, our deliverables were a GitHub repository where we injected some custom code, which you can use to train a custom model of this approach. And you can also use that custom code with a custom data loader to convert ATL training data sets to a colorized version of the dataset. We also did that so that you don't have to process the entire 90-something thousand 224 by 224 images. Unfortunately, Google Drive didn't let me paste the link here for some stupid reason, so I will share that with you as well. And a demo, of course, which I invite you to check out after the presentations. Thank you very much.
Really nice, Jaka.
Very nice.
Thank you. Also for the
Thank
demo,
you.
really cool. I think you managed to get some amazing results.
It's looking quite good, really. Actually, on the final sets that we ran it through, it does indeed do colorization. So that's quite nice.
Very nice. And also when working with multiple methods and also sometimes just the results not working out, very cool that you two pulled through and got these amazing results. And I'll sure be checking out the demo and play with it. And I'll definitely be amazed by the colors.
Undoubtedly.
Yes, I don't have anything much to add, but thank you very much, Jaka. I'm very impressed with the final results. I remember you did a lot of progress in the last few weeks, so that was very, very impressive. And yeah, I look forward to also checking out the demo. Thank you.
Thanks.
All right. Thanks a lot, Jaka. So I forgot to say that in the introduction, but if there are any questions during a presentation, feel free to put them in the chat. Or you can ask them right now if there are any questions about the color enhancement. And if they're not, yeah, if you come up with a question, make sure to put it in the chat. Then I think we can head on to the next group. So I'd like to invite Max on stage to share with us what he found on object segmentation.
Okay, hello everyone. I'm just gonna share my screen quickly. Is it visible?
Yes.
Yes.
Okay, yeah. So our team's goal was image segmentation. I will explain what that means in a minute. So we started with a bit more people, but in the end, we ended up with two. So it's me and Mark Halverson. He unfortunately cannot join today. He lives in Western Canada, so it's very early at his place, and it made our cooperations a bit challenging, but yeah, that's the team. So the problem of image segmentation, object segmentation, is figuring out what is where on the picture. So here we have an example with animals. So we want to have not only know what is on the picture, but what are the boundaries of the objects. And the goal of this is to apply this to the images of coral reefs to end up knowing the coral cover and like the proportions of how much coral is in a given area, how much algae. There were like 6 main classes that we were investigating. Yeah, and to calculate that, we needed to apply the segmentation. So our data set consisted of images of coral reefs with point annotations. So these were hand annotated by the divers, I think. So it took a lot of work to just annotate it. But the problem is that for segmentation, to train a model, normally you would start with hand annotated area annotations. So we would know like the shapes of everything. And here we had a bit of a more unusual kind of data with just point annotations. So we don't really know exactly the shapes of all the classes, but just approximately what points represent what. Yeah, we had to figure out some novel approach to this task. So the first thing we tried was unsupervised segmentation. So just ignoring all the labels, just trying to figure out based on the shapes of stuff, based on the colors of stuff, to try to segment it. But yeah, it was not good enough. And we thought that since we have the labels, we have to utilize them in some way to make it useful. Yeah, we also try to look for supervised data set with like area kind of labels or polygons. We search a lot, but it just is not out there currently. Maybe with our approach, we will be able to to actually provide such data for future researchers. So we were kind of lost in the beginning, looking for many different ideas and nothing working. But then when the coral classification team showed us some initial results, we thought that a good approach might be to just build a segmentation model on top of the classification model. Yeah, and that's what we did. So it's using classification model and simple linear iterative clustering. So this is an example of the SLIC, simple linear iterative clustering, which, as you can see, these orange lines on the picture, it divides an image into these segments based on the color values and the proximity of pixels. So it's a simple idea, but it does a relatively good job in capturing these complex shapes around the corals. And every segment has a central point. It's not exactly central for some weird shapes, so it's a centroid, which is approximately at the center. Here we applied the classification model. So you will hear about it more in their presentation, but I will just briefly summarize. So the idea is that they're able to take a small patch of this image, the square, which is like 200 pixels by 200, and provide a class for this, for the central point of the square. So based on this clusters, we cut out the squares around the cluster centroids, like here. So here are the centroids, and the classification team classifier is able to take a square patch like this and provide a class label. Is it a coral or algae or whatever? And then when we obtain the labels of the central points, we just assume that the whole cluster has the same label as the central point, which this is the result that we got from that. So here's an image, here is the segmented image, and here are the labels associated with it. As you can see, it's not perfect yet. So here this patch of I'm not sure if it's Coral or not, but it's clearly a uniform patch of something and it's not classified uniformly. But it has some idea of what's happening, and now like the performance is based on the classification model. Yeah, then we applied this this coverage calculation to a bigger part of the data set. We didn't manage to apply it on a full data set, but just on a part of the Indian Ocean data set, we got this kind of approximate results, which seem not that far from reality based on the estimations that we got from the Coral researchers. And yeah, there is still work to be done, but our approach has a couple advantages. So because we are using the model from the other team, we only need one model for the whole pipeline. And if we improve this model enough, we get classification and segmentation in one thing. And the sleek clustering provides fine-grained object boundaries. So like if the points are classified correctly, we can really finely estimate the exact area. And yeah, the clustering algorithm also is very easily adjustable. You can make more clusters, less clusters, and then you end up with either more precision or quicker runtime. And yeah, that's it from our team. Thank you.
Thank you, Max. Really cool. I do have to give you a compliment because your perseverance during this challenge was very admirable, especially during the last couple of weeks where there were a couple of setbacks in models that weren't working. I mean, every week you came out with new models, which was really good, and I really thank you for your hard work and your input. So thanks a lot about that. And I think for the results that you showed today are actually really close. So yeah, I think it's really nice, and I think the guys from Reef Support are very happy with you.
Yes, definitely. Thank you very much, Max, first of all, for the presentation, but also I have to say for seeing the challenges that you faced with the rest of the team. And I have to say, also very impressed with all the intuitive solutions you came up with every two weeks, new solutions to try out, and that was very impressive. I look very forward to seeing the end result, the code, of course, and play around with it. But thank you very much for the presentation, and unfortunately Mark could not be here as well, but thank you very much to both of you for all the hard work you've put in. Very impressive.
Yeah, to add on top of that, you've clearly explored many different types of machine learning as well. So it's not only like working with supervised models, but or only in the direction or concepts of CNN, but you work with both more classical clustering algorithms, unsupervised and supervised learning techniques. So I hope also that you learned a lot on that, and that now we all know that not always the most advanced methods work best. So thank you for learning us a lesson here as well, Max, and indeed really impressive results. This is also part of why Joppe and Stijn are here today from ML6. They will also be looking into this problem and seeing if they can even improve your results further. But I'm very sure that all the different things, the many different things that you tried will help them get along. And this way they will get a beautiful Christmas present to Reef Support as well. So that's make sure that your work is being pulled up as well.
All right. Are there any questions left for Max?
I was just going to ask, I guess the main challenge here was the fact that you didn't have like really clearly defined labels that were these annotation points, or?
Yeah, that was the main challenge, because normally segmentation model is trained with area labels or rectangles.
Right, that's an interesting challenge to tackle indeed. Yeah, well done.
Okay. And I think we can kick off the last presentation then. We have Leonard here, and he will tell us everything there is to know about classifying corals.
Hi guys, let me just quickly share my screen. Okay.
Yes, we see it.
Okay. You're seeing the presentation screen, right?
Yeah, it's good.
Perfect. Let's see that it's not in the way. Okay, so yeah, let me just quickly introduce our team. So our task was basically, well, Max already explained part of it, that we try to train an algorithm for coral classification. And the remaining team, I hope I didn't forget anyone, is seen here underneath the what's it called, the acknowledgements. And as you can see, we're quite a diverse group, different backgrounds, but in the end, this was probably also an advantage for us. Okay, so I mean, we touched on aspects of this already, but ultimately the goal of classification of coral reef benthos is, of course, the big promise of making large-scale automated prediction of what is there, basically. And we had, of course, to aim a little bit lower. So our minimum goal in the end was basically to classify coral from non-coral, including hard and soft coral, probably. And we ended up somewhere in the middle, where we chose, well, I can skip this probably, where we chose from the functional group annotations that were made or that came along the Caitlin Seaview survey image data set that we had. And good about this data set was that it's all taken with the same method, probably slightly adjusted over the years, but so we get this image squares, which are approximately, I guess, a square meter or so, around 1 million pixels each image. And yeah, so there was some uniformity in that. And I think we had this picture already in Max's presentation as well. So those pictures, which came in, I think, yeah, they are almost a little more than 10,000 images, came with annotated point labels for, I think, 6 different functional groups. And they're under stuff or benthic organisms such as hard coral and algae. Quite a complex surface, as you can see. So not an easy task. And basically, the first thing we had to do was to create from these 50 points per image, 40 or 50 points, pixels, so to speak, in 1 million pixels to create a more dense labeled image as ground truth, basically. And yeah, this is just the data set format which it came from. So we chose this functional group instead of going even deeper into coral species or stuff like that, which is well, probably a couple of way more difficult. Yeah. And yeah, so one thing we didn't really dealt with, but we had to acknowledge, was that we had a slightly imbalanced data set. So I think this was this chart here is basically a representative of the Atlantic data set, which is, I think, 1,400 images or so. You can see that most of the labels correspond to the algae functional group, which comes almost in a relation from 5 to 1 to the ones that are really interesting to us, which are hard coral and soft coral. Yeah, but we decided not really to deal with that at the moment, but to keep that in mind for sure when looking at the accuracy of the model, for example. And this could also be a motivation to look into further data augmentation for other groups to create statistical data to match the different classes to get a more balanced data set. Yeah, so since the input format for most algorithms came in the format 224 times 224 pixels, we had to well, crop these point labels or around these point labels in a uniform fashion and basically create, yeah, those crops, 50 for each image. And as you can see that this is probably labeled sand or substrate or something like that. If that point would be in the middle of the coral here and labeled as hard coral, then the majority of pixels would still be from a different class, well, which is part of the intricacy of yeah, of this task. And exactly, and those were basically the input images that we fed into our algorithm in the end to get first and foremost just accurate predictions of the different classes that we chose. Yeah, so here could be algae instead of also coral or something else. And like that, we created our dense labels basically. Yeah, throughout the process, throughout the weeks, we tried a couple of different neural networks and vision transformers, for example, mostly pre-trained on the ImageNet set, which is of course not a benchmark set for coral reefs, but still a quite extensive collection of images. And we didn't really get any results in the beginning because we had issues dealing with such large amounts of data until we decided to actually yeah, work with the fractions of the data set. But we started to try just to create NumPy arrays basically and save those in a big file as in what's it called? I'm not sure, but the H5PI package or format provides this possibility. And like this, of course, we maxed out our memory quite quick. So Path instead came up with just using a PyTorch script with the dataset class. And like this, with this custom dataset, we were able to basically process the images and crop images on the go. And we used it in combination with basically the GoogleNet, which is based on Inception architecture. And this also is, well, in terms or in comparison to the VGGs, for example, a quite economic network and probably better fit for low-end technology like we're using. And yeah, the only thing that we had to do there was, or the big one is, of course, that the images we put into the algorithm had to be normalized according to the ImageNet data, but that wasn't a big deal in the end. Yeah, so we used this pre-trained model basically and only fine-tuned the last fully connected layer in the architecture, and it achieved, I think, after just 2 epochs already. So this is after a couple of setbacks, of course, quite a good overall accuracy, above 80%. And we basically use those weights and the optimizer function, which were, I think we just used the default one. I'm not sure at the moment. And just run this again for a training of 4 epochs in total, basically, to achieve around 86% accuracy on the classes. So for those of you who don't know what this confusion matrix is telling you, is basically in the diagonal we have our 6 classes. The interesting here are, of course, the hard corals and the soft corals, which were quite underrepresented in the data set in comparison to the algae class, for example. And the x basically tells you the predicted label of the model in comparison to the ground truth. Yeah, so you can see that also here in the other metrics. Yeah, to what degree the model were able to predict. So if you can transform that into percentages of images that were fed into the algorithm, the right labels. Yeah, this is basically the state of our results at the moment. I mean, we also try to calculate the coverage from that, at least for the hard corals and the combined hard coral and soft corals, but those ended up to be quite a, I think, overestimation. It was in the end of the actual, data we had about this specific data set about the Atlantic. But I mean, this is something, to be fine-tuned and worked on in many different ways, I guess. And for that, we come basically to the final slide, which is the recommendations. Of course, one thing to fine-tune this is approach syntactical data to balance the data set and also to optimize, for example, image borders. I think also Max did something like this and just mirroring the last couple of pixels before the border or something like this. There are also indications that a multi-scale input format could help the algorithm. So not using or not relying solely on 224 to 224 dimensions. Or it's a shame that we didn't talk about this earlier, but to work more with those superpixel approaches also that the object detection group worked with. Other interesting things, well, it's just a practical issue that we encountered just because we also had other different data sets from other organizations. And of course, stuff like labeling and selection of classes were, of course, not uniform across the different organizations. So this is a data sharing issue of global dimensions, basically. And well, I mean, there are also other ways to approach this that could provide more feasible approaches, also in combination with citizen science, instead of, for example, trying to go for specific species, just set back the resolution and decide on certain habitat types or predominant organisms in the benthos, stuff like that. Yep, that's basically where we ended up with that challenge, conservative speaking, but I'm sure we all learned a lot, and yeah, thank you so far.
Thank you, Leonard. It's great to hear that you also learned a lot. I think you managed to get some good results as well. And I already got a feeling that we're just 2 weeks short of really coming up with all the exactly the right solutions that we aim for. But on the other hand, still some nice stuff left for the ML6 people. But I think you did a really good job, Leonard, and thanks for all the hard work for your whole team, of course. So thank you.
Yes, thank you very much, Leonard, and of course to your whole team. I have to say you were all very well organized from the get-go. You had a clear idea of what you wanted to do, and I was very happy with your team in terms of how you divided the tasks and how you went about tackling the challenge. I'm very happy with the end result, and I also look very forward to trying it out myself, your implementation. So yeah, thank you very much for your presentation, and thank you for the 2 months of hard work you put in.
Yeah, indeed, Leonard. Also from my side and to the whole team, of course, amazing results. You already got very decent accuracies early on, but you have been improving on those all over the time. So I also hope that you learned a lot indeed. But I also had 2 questions, actually. So I had contact with one of your teammates about different types of models that you have tried, and you also have been looking into various visual transformers. What is your experience actually on that, and how they compare to the CNN-based inception model that you ended up using?
Yeah, maybe Path can elaborate on that a little bit, because he especially had a personal relationship to those vision transformers. I don't know what the state of this was in the last weeks, but I think you recommended those, right, Sako? Or...
Yeah, so at some point I got some questions about that, and then I think Path indeed tried out a few of those fishing transformers and also got decent results, as I understood. So I'm curious about the comparison, basically, your experience, because this is quite a relatively new technology. Transformers on the one side, but especially using it for vision tasks. So insights in that and why you ended up with the inception-based CNN model is of course very interesting to share with everyone.
Sure. So I mean, a lot of decisions were just made out of conveniency or out of being convenient, or out of experience, of course. So I mean, would be nice to have a lot more time to experiment, or even like align models with other groups. I think Path will join us in a second.
Oh, he will update us in the chat.
Okay.
So in the meantime, it's good that I had two questions. So the other two teams have been working on stuff, of course, data set enhancement in some way. So on one side, it is color enhancement, which ended up working pretty well, and also the segmentation. Both of those could, of course, help towards also classifying better, right? So if you would have decent segmentation of the corals, you could get better centroids for your cutouts for the classification. And the same, of course, holds for color correction, which would, well, increase the contrast. Do you think, have you been able to test if any of those things helped your classification? And do you think, and what is your view on how that would have implemented accuracy?
Yeah, it's a shame that we didn't really get to that part. Sorry on that. So we got some images from Yaka, like he told you, but in the end, or the only try we got, we kind of missed because it seemed like the transformation, the images went through a mess with our algorithm, so our format we used, and we didn't really were able to sort out that problem before this week. But I mean, somebody should for sure, try to look into that.
Yeah, from our side, we were a bit late, unfortunately, for providing a revised version of Color Enhanced Dataset, and I will still have to check their data loader to adapt it to the output, because it is a bit different than just immediate plug and play. And then you guys can give it a try as well.
Very cool. I'm sure Johan will, and then give us an update, Johan, what the accuracy differences are. But you think, Leonard, as well, that these color enhancement, including also better bounding boxes, can increase the accuracy a lot, or do you expect it actually to stay around this?
Well, I don't know if it's just the color enhancement, but we had, I think, like intermediate results also that took into consideration just contrast or something like this, or texture. So I'm sure there are other approaches as well. And yeah, I mean, I know also that there are studies out there that work, for example, with the UV light to get the fluorescence of the corals, which of course has a way different picture and contrast, in terms of the input channels, which also can help. But I mean, yeah, I've seen the images at least. Don't really know how it worked out in the end, but I guess this could help, yeah, for sure.
Okay, very nice. Well, thank you to the classification group, all of you, for your hard work, and of course, still to all the teams. I think there are some very cool results that Reef Sport will be able to help the world of coral reefs with. So that's, of course, why we're here, right? So that's amazing that we can get to these results.
Yes, yeah, thank you very much to all the participants and all the three groups. I think I speak for both Marcel and I, and Jasse is also listening here, but I think I speak for all of us at Reef Support when you say that we're very happy with the end results. And of course, big thanks to you, Sako, and Dorian, and Buster, who's not here unfortunately, but yeah, this challenge has really been amazingly well organized, and it was a great opportunity for us as well to work together with more experienced AI enthusiasts all over the world and share our idea as well. So it's really an amazing opportunity. Thank you very much for that. And again, thank you all very much for the participants.
Thank you, Johan. Yes, I have to say it was a pleasure working together. And I really have to give a big thank you to all the participants and everyone who put in the extra hours after work or after study time, all towards a better and sustainable future, and to conserve the coral reefs. And I think we did a really good job. So I believe that everyone can be very proud of himself and herself. And yeah. We had all the presentations here tonight. I hope everyone who wasn't part of the challenge and was here to watch enjoyed themselves and understood everything. The recording will be posted on YouTube if there are things that you would like to rewatch. All our end results will be open source as well. So if you're interested in all the results and want to have a more detailed look into what all the teams has done exactly, then that is possible as well. You'll be updated. So keep an eye on the Fruit Punch and Reef Support LinkedIn, but also the Fruit Punch platform. All right, then I'd like to thank everyone for being here, and I wish everyone a very good night.
Yes, thank you very much, Dorian. I don't know if it's good night for everyone, but otherwise enjoy the rest of the day, and thank you all for being here.